Welcome to Decoding Autism Now, where science meets clarity about autism. I'm Dr. Elise Martin, and this is an expanded version of Episode 9 from our series based on the book Understanding the Rhythm of Digestion in Autism by Stoner. This will be a deeper dive into one of the mysteries about autism: why boys are diagnosed nearly 4 times more often than girls. This is not simply about diagnostic bias. Biology is speaking, and when we listen closely, we find a fragile enzyme at the center called Adenylyl Cyclase. To understand how this fragile enzyme works, we must follow the cascade backward, because autism's story does not begin in the brain. It begins in the gut, with digestion. And it starts with something as deceptively simple as stomach acid. When this fragile rhythm falters, proteins remain undigested, peptides escape into the bloodstream, neurotransmitter supply runs short, and the brain loses its beat. This is the first domino in autism's gender puzzle. This episode is both a recap and a deep dive. Together we'll retrace key insights from chapters 1 through 8 of the book Unlocking the Rhythm of Digestion in Autism by Stoner, Setting the Foundation. Then we'll focus on chapter 9, and what I call Autism's Gender Puzzle Decoded. Why are boys diagnosed so much more often with autism than girls? Could the answer lie not in culture or social bias, but in a fragile enzyme called Adenylyl Cyclase and the protective role of estrogen? By the end of this episode, you'll see how something as simple as stomach acid can ripple upward into neurotransmitters, hormones, and finally converge at a cellular crossroad inside every human cell. And this may explain one of autism's biggest mysteries. Could this be the first step in understanding the cause? Can this be the key that allows us to move towards a cure? Stay tuned as this gets very interesting. Let's begin at the very start of digestion, the stomach. Before food even touches your lips, your body is already anticipating gastrin signals from the parietal cells. This starts the release of hydrochloric acid, while secretin and motilin stand by, waiting for the right pH cue to set the next stages of digestion into motion. But here's the catch: this cascade is fragile. When stomach acid production is too low, a condition called hypochlorhydria, the dominoes fall out of rhythm. Proteins are only partially digested. Gluten and casein fragments linger, forming opioid-like peptides. Secretin and motilin fail to fire on time, leaving pancreatic enzymes and bile unreleased. Acid is more than chemistry. It's the conductor's baton of digestion. At a pH below 2.0, pepsin is activated, slicing proteins into amino acids. But if stomach pH drifts higher, even slightly, pepsin goes silent. And with pepsin offline, amino acids like tryptophan and tyrosine, the building blocks for serotonin and dopamine, remain locked away. This is why children with low stomach acid may eat enough protein yet remain biochemically malnourished. Their pipeline to neurotransmitter synthesis is blocked at the very first step. When acid falters, microbes seize the chance to overgrow. The upper intestine, normally acidic and tightly regulated, becomes a playground for bacteria. This condition, called small intestinal bacterial overgrowth or SIBO, sets off a storm. Carbohydrates ferment too early, producing hydrogen, methane, and toxins. These microbial byproducts irritate the gut lining, leading to inflammation and permeability. Undigested peptides and bacterial fragments cross into the bloodstream, where the immune system flags them as threats. Cytokines rise, and cytokines don't stay local, they travel, reaching the brain. The result? Heightened microglial activation, mood volatility, disrupted sleep, and sensory hypersensitivity. One enzyme sits at the center of this drama, CD26, also called DPP4. Its job is to serve as a binding site for adenosine deaminase, the enzyme that breaks down adenosine. If we accumulate too much adenosine, it leads to being tired, and more importantly, it slows down the methylation cycle. But this receptor can get clogged by opioid peptides. These peptides are the partially digested proteins found in certain grains and dairy. These opioid-like peptides can also the blood-brain barrier, creating narcotic-like effects. Children may seem dreamy, emotionally flat, or locked in repetitive behaviors. Others may show strong cravings for bread or cheese, feeding the cycle. Proteins are the scaffolding of life, the raw materials for enzymes, hormones, neurotransmitters, and even immune receptors. But with poor digestion, these proteins never reach their destination. The result is hidden malnutrition. A child can eat enough calories and protein yet remain functionally starved. Low tryptophan means low serotonin, fueling anxiety, mood instability, and sleep disturbances. Low tyrosine means weak dopamine and norepinephrine, contributing to low motivation and poor focus. Methionine shortages impair methylation, slowing detox and DNA regulation. Zoom down even further, and we see disrupted calcium signaling inside neurons. Calcium ions normally enter the cell in rhythmic pulses, tiny sparks that activate genes and trigger neurotransmitter release. Without adequate amino acids, these calcium puffs misfire. Genes switch on and off at the wrong times. Neurotransmission becomes noisy instead of clear. This isn't just malnutrition, it's miscommunication at the cellular level. And here we begin to foreshadow the fragile enzyme we'll explore later in the video, Adenylyl Cyclase, which itself depends on calcium pulses for activation. Now let's revisit those gluten and casein fragments. When not fully digested, they form exorphins, gliadorphin from gluten and casomorphin from dairy. Exorphins are molecular mimics of opioid drugs. They slip across the leaky gut, bind to opioid receptors in the brain, and hijack the reward system. Children may crave carbs and dairy like an addict craves their next dose. After eating, they may appear calm but detached, their focus blunted by narcotic-like fragments. But the damage isn't only behavioral. Exorphins clog CD26, blocking the enzyme adenosine deaminase. Without ADA, adenosine builds up, slowing methylation and overwhelming detox. Elevated adenosine overstimulates ADA receptors, driving oxidative stress and neuroinflammation. The immune system falters too. T cells with opioid receptors are suppressed, leaving the child vulnerable to chronic infections. Parents often describe a cycle: dietary slip, infection flare, behavioral regression. That's the peptide trap in action. Your body is more like an orchestra than a machine. Hormones and enzymes play their notes in rhythm, and when they fall out of sync, the entire piece collapses. Our stomach acid sets the tempo, like the conductor's downbeat. Gastrin, secretin, motilin, and CCK pulse in carefully timed order, cueing enzymes and bile. The vagus nerve acts as the baton, linking gut rhythms with brain states. Even inside cells, calcium sparks create a rhythm for neurotransmitter release and gene activation. When rhythm falters, digestion becomes chaotic. Neurotransmitters dwindle, sleep cycles break down, anxiety and sensory overload take over. Clinically, we see children with erratic energy who oscillate between hyperactivity and fatigue. Some show food-seeking patterns that resemble cravings more than hunger. Others have nighttime waking as hormones like melatonin fail to sync with digestive cues. The key message: autism is not random chaos. It is disrupted rhythm, biochemical instruments out of tune. Restoring rhythm through timed meals, vagal exercises, and reducing stressors can help to reset the score. By now, we've seen how low stomach acid disrupts digestion, nutrient absorption, and rhythm. But the ripple effects go even further. With hypochlorhydria, the stomach's pH rises above the threshold where pepsin can function. Proteins remain intact. Gluten and casein peptides slip into circulation. Zinc and B12 are poorly absorbed, and the gut's microbial balance tilts. Now, let's zoom in on a receptor called CD26, the same enzyme we saw earlier being blocked by exorphins. Low stomach acid makes CD26 even more vulnerable. In fact, it faces what I call a triple threat. First on the list are the peptides gluten from grains and casein from dairy. They bind to a receptor on CD26, blocking the ADA enzyme needed to break down adenosine. A second item is heavy metals like mercury or lead attaching to its active site. And finally, the third is streptococcal toxin, common in kids with chronic infections or PANDAS, binding and suppressing its function. When CD26 is jammed, adenosine deaminase, ADA, can't work. Adenosine builds up in the system like exhaust fumes in a closed garage. The result? Methylation, the body's master regulation cycle, slows down dramatically. DNA expression falters. Neurotransmitter synthesis weakens. Detoxification bottlenecks. High adenosine overstimulates ADA receptors, which crank up oxidative stress and neuroinflammation. Parents notice the fallout: restless sleep, emotional volatility, sudden regression after illness, and an almost allergic-like sensitivity to environmental changes. This is not a minor hiccup in digestion. It's a biochemical choke point where diet, toxins, and microbes converge to derail metabolism. Now we arrive at a powerful hormone that acts like the body's emergency brake: somatostatin. The job of somatostatin to turn off acid production when digestion is complete. It shuts down acid production, slows pancreatic enzyme release, and conserves resources. But in many children on the spectrum, somatostatin is chronically elevated, pushed there by stress, inflammation, or the flood of opioid peptides we've already discussed. When this hormone gets stuck in the on position, the consequences are profound. Acid and key enzymes like secretin stay suppressed long-term. Growth hormones and thyroid signaling weaken, stunting physical growth and slowing metabolism. Neurotransmitters like serotonin and dopamine dry up, leading to mood instability, fatigue, and withdrawal. Just a side note that you might want to check out episode 2 on this channel titled Why Digestive Hormones Matter in Autism. In that episode, I discuss an accidental finding about secretin by a parent over 30 years ago. It has now been buried by time, but it is very relevant today in understanding autism. I urge you to review that again so that it can get the attention that it deserves. Overexpression of somatostatin essentially locks the body into survival mode. The system stops repairing, regenerating, and growing. Clinically, this shows up as stalled milestones, regression, or children who seem developmentally frozen. Even the immune system is distorted. Somatostatin can calm inflammation in the short term, but when it is overexpressed long term, it leads to a suppressed immune function. The result means children are left vulnerable to recurrent infections. Think of somatostatin like an emergency brake. It was designed for temporary control, but when pulled constantly, it halts forward momentum and damages the engine. So far, every chapter has pointed to fragility in digestion, immune regulation, and neurotransmitter balance. But all these loops converge at one deeper level, the way cells themselves translate signals. At the cell membrane, G proteins pass messages from receptors to an enzyme called Adenylyl Cyclase. And Adenylyl Cyclase generates CAMP, the universal signal for neurotransmitter release, hormone action, energy balance, and immune regulation. It tells the cell what it needs to do. Here, at this fragile switchboard, lies one of autism's biggest mysteries: why boys are so much more vulnerable than girls. Autism is diagnosed in boys nearly 4 times more often than girls. Traditionally, this gap was explained by diagnostic bias. Girls mask symptoms better, or clinicians are less attuned to female presentations. But biology tells a deeper story. Estrogen, the primary female hormone, provides protective detours in cellular signaling. Girls are born with higher baseline estrogen exposure, even before puberty. This buffering allows them to reroute around weak adenylyl cyclase activity. Boys, however, rely more heavily on dopamine and androgen pathways for a very small amount of estrogen, which can funnel through AC. When AC stalls, boys lack alternative routes. They're left more vulnerable to regression, mood instability, and fragile development. We often hear the word estrogen, and our brains think, that's the female reproductive hormone. But it is far more powerful because it is a protective hormone and a signaling hormone. At a conference in 2004 while giving a presentation, Dr. Jill James spoke about a study her group had performed. They were testing the impact of a preservative in vaccines. Glioblastoma cells found in the human brain were used to test their survival rate when exposed to the preservative. Half of the test cells were bathed in estrogen, and the other half were not. The cells bathed in estrogen survived 10 times longer. This exercise proved the protective properties of estrogen. Clearly a watershed moment in the lab about something seemingly small. This difference, protective redundancy in girls versus single-point fragility in boys, may explain the striking gender ratio we see in autism. So, at this point you are probably asking why something so obvious has been ignored for so long. Stay tuned because we're about to couple that one small lab event with something else that has been flying under the radar for a long time. And when combined, these two events yield something so exponential that it could change what we know about autism. Before I continue, please like and share this video with others. Your small action of sharing can impact the lives of others. Please subscribe for upcoming episodes on the channel. That helps the YouTube algorithm make our channel visible to others who also have an interest in autism. Let's take a closer look at adenylyl cyclase, or AC for short. Think of AC as a translator. When a neurotransmitter binds to a receptor, the G protein carries the signal to AC. In turn, AC converts ATP into CAMP, the messenger that tells the cell what to do next. CAMP is like electricity in a circuit. It drives several hormones we need for additional signaling in our body. Dopamine release is necessary for attention, reward, and motivation. Serotonin signaling helps with mood stability and sleep. Oxytocin release improves our bonding and social connection. CAMP signals our immune system to turn inflammation on or off. It has an impact on energy metabolism, helping to keep the mitochondria running smoothly. But when AC stalls, it also stalls CAMP. The CAMP circuit goes dark, and our dopamine falters, resulting in poor focus and reward seeking. Our serotonin weakens, which leads to mood instability, anxiety. Oxytocin lags, and that leads to disconnection in social interaction. Our immune control slips, leading to inflammation flares. The body's energy is depleted because of the low ATP conversion in the citric cycle. This is why stalled AC can feel like 1,000 symptoms throughout our body, but only one thing is responsible. It can manifest as cognitive, memory, touch, gross and fine motor skills, balance, emotional, taste, smell, OCD, and so many more symptoms. It isn't just a brain problem, it's systemic, touching every cell where CAMP is in short supply, impacting function. As we discussed a bit earlier in this video, estrogen is far more than a reproductive hormone. It's a neuroprotective signal that can bypass adenylyl cyclase. Think of estrogen being able to hotwire around the broken AC where it can activate CAMP. And this is where we connect two seemingly unrelated topics of estrogen and adenylyl cyclase. When these are viewed together, it provides a totally new perspective. Estrogen bypasses the broken AC signaling link in the chain. It causes ATP to be converted to CAMP, the second signaling molecule. In girls, this keeps the signal moving so the cell knows how to react and what to do next. This enhances serotonin release, promotes neural growth and plasticity. It keeps the signals functioning in the brain. It shields neurons from oxidative stress. And most importantly, it can bypass weak AC signaling to keep CAMP's second message signal working in over 20 trillion cells that are in our body. This is why girls often show resilience. Their brains have estrogen-mediated detours that stabilize signaling. Even in the face of toxins, peptides, or genetic variants, estrogen provides an alternative biochemical pathway. Boys, by contrast, depend on straight-line signaling through AC. Without sufficient estrogen buffering, they're left exposed. This vulnerability may explain why regression, hyperacusis, and mood swings often hit boys harder. Estrogen doesn't eliminate autism, but it may reshape its severity and expression, a powerful clue to why biology, not just bias, drives the gender gap. Why does adenylyl cyclase stall more often in autism, particularly in boys? The evidence points to multiple overlapping hits. First, toxins and heavy metals. Mercury, lead, and aluminum can bind sulfhydryl groups, interfering with G-protein coupling and AC enzyme activity. These toxins also deplete glutathione, making neurons more vulnerable to oxidative stress. Second, opioid peptides, gluten and casein fragments block upstream receptor activity. When the receptor to G-protein message is distorted, AC receives a weak or scrambled signal. Third, chronic inflammation. The kynurenine pathway diverts tryptophan away from serotonin and toward quinolinic acid. Quinolinic acid is an NMDA receptor agonist that floods neurons with calcium. This toxic overactivation burns out AC responsiveness, leaving the enzyme less able to translate signals. Fourth, genetic variations. Certain AC isoforms have polymorphisms that reduce their efficiency. These genetic differences can magnify vulnerability when layered on top of environmental stressors. For boys, without the buffering detours that estrogen provides, these insults accumulate. AC fragility becomes not just possible, but predictable. AC, or adenylyl cyclase, isn't a single enzyme. It's a family of 10 isoforms, each with unique roles. AC1 and AC8, brain-based, vital for learning, memory, and sensory processing. AC2 and AC4, integrators, balancing inputs from multiple neurotransmitter systems. AC5 and AC6, heart and adrenal regulation, controlling rhythm and stress responses. AC7, immune modulation, balancing cytokine release and inflammation. AC9, fine-tuning signals in the airways and gastrointestinal tract. AC10, soluble AC, critical for mitochondrial energy and sperm motility. When AC isoforms are unstable, the fallout is wide-ranging. In the brain, there can be sensory overload, hyperacusis, learning struggles. In the heart, impaired cardiac AC isoforms can cause palpitations, poor stress recovery. When immune AC isoforms falter, the conditions can be chronic inflammation and impaired autoimmunity. Gut AC isoforms are responsible for motility issues, constipation, or diarrhea. And when our mitochondrial AC isoforms are not working properly, we see fatigue and low energy. This paints autism not as a brain-only condition, but as a system-wide signaling fragility. The same weak enzyme ripples through learning, immunity, metabolism, and more. How can we recognize stalled adenylyl cyclase in practice? While no single test yet exists, clinical patterns offer clues. One clue might be fluctuating levels of neutrophils and eosinophils. These are generally linked to inflammatory responses and are often included with blood test panels. Fluctuations could be due to bacterial infections, stress, tissue injury, systemic or chronic inflammation, autoimmune changes, allergic reactions, viral infections, histamine levels, and more. Consult with your medical professional for more information. A stool analysis is another medical test that can be used to identify if there are issues. Results can show the pH levels in the intestinal tract, if parasites or other organisms are present. It provides details about digestion. If the stool diagnosis shows gut dysbiosis, that is clear indication things are not right with digestion. There are labs that will test the urine for undigested proteins known as peptides. Having urinary peptides is a sure indication that proteins are not getting broken down by digestive enzymes like pepsin. I have a theory that excess saliva may be an indicator. Saliva is rich in the enzyme amylase, which helps break down starches even before they reach your stomach. Having excess in the form of drool may be the body getting a head start on digestion. Again, this is just my unproven theory, but it is backed by science. Sensory issues are another sign in autism. It can manifest as fragility with touch, visual overload, acute hearing for loud sounds, certain smells, and more. These can be more noticeable during inflammatory episodes or too much stimulation. Mood instability and cyclical emotional swings can be linked to infection, stress, or even weather changes, all of which influence inflammation and signaling. There might be a fight-or-flight response, and that can be due to overworked adrenals trying to push the GTP signal into the cell, which we covered in previous episodes. There is female resilience, where girls show a surprising ability to bounce back after stressors, hinting at estrogen buffering. These aren't diagnostic by themselves, but they form a constellation pointing toward AC as a fragile signaling hub. It should be much clearer to you that AC is the common item for issues with sensory, cognitive, learning, memory, and more. There is no single therapy to fix adenylyl cyclase being able to pass a signal. But we can create conditions that make the signaling more resilient, supporting the system around it. Let's review the basics, and as I covered in other videos on this channel. First, address the foundational issues. Nutrient cofactors such as magnesium, B vitamins, and zinc are essential raw materials for CAMP production. Without them, AC cannot sustain its function. Then there are the categories of polyphenols and flavonoids. These are compounds like curcumin, resveratrol, and EGCG. These can stabilize estrogen pathways, help calm inflammation, and reinforce fragile signaling loops. And after addressing the foundational basics, consider these three emerging supports that are worth highlighting. Forskolin, derived from the coleus plant. Forskolin is one of the rare compounds that directly activates AC, boosting CAMP production. This can help unstick stalled signaling, though it must be used carefully under medical supervision due to its cardiovascular effects. Saccharomyces boulardii, a probiotic yeast that reduces gut inflammation, crowds out harmful microbes, and lowers toxin load. By stabilizing the gut, it indirectly protects AC from inflammatory overload. Genistein, a phytoestrogen with antioxidant properties. Genistein offers boys a biochemical detour by partially mimicking estrogen's protective effects, helping buffer signaling when CAMP production falters. Genistein is non-feminizing and can be from non-soy plant material. Together, these strategies don't override biology, they nurture it. The goal is not to force signaling, but to create a supportive environment where fragile pathways can flow freely. As always, you should not make any changes to your health routine without first consulting a medical professional. These suggested strategies should be shared with your medical professional, so they can provide the proper guidance for your specific situation. This is also a call to action for medical professionals about the need for more research of adenylyl cyclase and estrogen as they relate to autism. There has been research on each individually, but studies are needed to review these in concert. By now, it is becoming more obvious how they shift the perspective for autism. The gender puzzle in autism is not just a social artifact, it's deeply biological. Estrogen provides protective detours. boys with less estrogen buffering are more exposed when AC stalls. The gut, peptides, tryptophan pathways, and immune stressors all converge on this fragile enzyme. By seeing autism through this molecular lens, we move past stereotypes and begin to understand why boys so often show higher prevalence and more severe forms. To recap this journey, we traced how weak stomach acid and peptides derail digestion. We saw dysbiosis, hidden malnutrition, and somatostatin lock the system into survival mode. We followed the trail to a fragile signal at Adenylyl Cyclase, the enzyme that translates cellular communication. At the AC link, we uncovered how estrogen can keep the cellular signaling alive for girls, while boys lack this biochemical option. We discussed the lab experiment proving that estrogen can protect neural cells from insults. We explored ways to reduce inflammation and stabilize signaling pathways. And we discussed what can be added to help the cellular voice be heard more clearly. The lesson is clear. Autism is not simply a behavioral condition. It is a signaling disorder, a cascade of fragile pathways that ripple from gut to brain to hormone. And that can lead to a spiraling state of health. And here is the hopeful part. Fragile does not mean broken. With the right supports, nutritional, microbial, and hormonal, signaling can be steadied. Resilience can be restored. I'm Dr. Elise Martin, and thank you for joining me for this episode of Decoding Autism Now. If you learned something new, please like this video below and share it with parents, clinicians, and researchers who may benefit. Don't forget to subscribe so you don't miss upcoming episodes, where we continue making the invisible visible. Final thought. Sometimes the smallest stalls in cellular communication can ripple into life-changing outcomes. By restoring flow at the molecular level, we open doors to growth, stability, and connection.